Thank you, Mia, for that kind introduction. So what I would like to talk to you about is a little bit of the evolution of this program, as well as evolution in a biologic sense and its relevance to undiagnosed diseases. So I've had the best of jobs that one could imagine at NIH because fundamentally I was given a sandbox and said develop concepts. As you would think about this for the clinical practice here or elsewhere, what do you do when a single patient comes in with a disease that you don't recognize? You don't have a cohort. You can spend a long time trying to build a cohort, but if you're not going to do that, how do you go forward? What are the concepts and the issues that you need to deal with? And that fundamentally was the issue for which I was hired by Dr. Gall in 2010 to try to address these problems and try to figure out a way forward. As you can imagine, as this expands into the extramural environment, each institution is going to have to grapple also with these issues and how you try to go forward. In many ways, you could say, medical genetics clinic is that, or a tertiary care center anyway, because you're at the end of the referral apex. So the Undiagnosed Diseases Program was founded in May 2008. It's at that time, it was an intramural trans-NIH program, and it had two objectives: to provide answers to patients with mysterious conditions that have long eluded diagnosis, and to advance medical knowledge. When Dr. Gall called me and asked me if I was willing to work in this program, I asked him for the business plan, because I was quite curious why many intelligent people would be willing to invest millions of dollars in a diagnosis. You notice there's no statement here about treatment, therapeutic outcomes, and any of that. And, and he said, well, there wasn't one. We thought it was a good idea. So what is a diagnosis? And this came to the fore when I received this contact from Beijing Genomics, and they said, could you set up an undiagnosed program in China and give us some advice on that? So I scratched my head and said, well, how do you know the diagnoses we are giving are in any way relevant in China? And that had strangely never crossed the minds of the people at NIH, that the diagnoses we give here are irrelevant in China. They don't make sense, because diagnoses are cultural constructs. So in China, if one went into traditional Chinese medicine, no one lacks a diagnosis. Everyone has a diagnosis. So there isn't an undiagnosed disease in China. So why would the program exist? Where would the patients come from? Anyway, interesting to think about, because it does raise some issues as you would consider developing an undiagnosed diseases program, that what you would develop is going to appeal to some people, but not others. The reason for a diagnosis is that it provides meaning and trajectory for life. It gives people some sense of where they're going, or their child is going, or their loved one. Within the West, in the Occidental thought, we've moved dramatically in what we consider reasonable diagnoses over the last millennia. From mystical or theocratic diagnostic explanations to more rational diagnoses. And this latter arises or comes to increasing prominence after the Enlightenment, and in philosophical terms is considered scientific materialism, or reductionism probably is a less loaded word. Anyway, within that context, of these occidental reductionist diagnoses is where the UDP is parked. And that's where the extramural diagnostic UDP programs would also be parked. So, as you know, the Undiagnosed Diseases Program funding ran out a year ago, August. It was decided to refund it, but with the expansion to 5 to 6 extramural sites, and use this in addition to the CTSAs as a model for translational medicine. So what I want to do now is move through some concepts that have been very useful to formulating the Undiagnosed Disease Program and actually have anchored what we do and how we can move forward scientifically. Some of this is pretty obvious, but we don't really think about it very often. Or at least that's been my impression speaking with the people at NIH. So, the first is, in evolutionary context, each human is the product of a unique evolutionary history. That means every human has a unique genetic code. Each human is the product of sex, and therefore has their own set of recombinations and mutations arising out of that. Second, each human is the product of a unique developmental, cultural, and environmental history. This has some fairly profound implications when you can begin to consider the analyses and how we approach trying to diagnose these undiagnosed or undescribed disorders. This means that health, then, is adaptation to an ecological niche, and disease is the converse. Thinking about that for a second, you realize common disorders are not genetic. They are merely expressions of genetic vulnerabilities. They are changes in the ecological niche. So trying to study obesity from the perspective of genetics is absurd. You aren't going to get anywhere. I mean, you can consider the human population as it exists today as a nice mouse colony in which you're going to find, sensitivity and resistance alleles, but it's an absurd genetic concept. Now, for indirect costs and funding from NIH, it's a very good concept. But it's not scientifically sound. The other thing that follows from this is disease can never be eradicated. Some people who talk about curing disease or eliminating it, it's just, I don't know, it doesn't work. If humans are living and humans are the product of sex, then there always will be disease. It is part of the being alive. It is part of the intrinsic variation that makes each of us unique and makes each of us human. This, I'll emphasize again, common disorders arise from alterations in the ecological niche and have low heritability. As you correct the ecological niche, the heritability will rise, and the remaining individuals with problems, after you remove the ecological components that are exposing the vulnerability of the general population, those individuals will have a strong genetic component leading to whatever the problem you're considering is. Also, that means that if we want to treat as part of our objectives, we are going to be best at prevention. And for those that we cannot, disorders that we cannot prevent by changing the ecological niche, there's only two therapies: change the ecological niche of the person, or improve their adaptation. And there's no aspect of medicine that is exempt from this, whether you're a surgeon or whether you're a geneticist. Surgeons are improving adaptation, so they are functioning within a genetic context. Everything they do is subject to a genetic interpretation. From our perspective as medical geneticists, what's easier if somebody has ectodermal dysplasia? Allow them, try to correct their ectodermal dysplasia and their predisposition to heat shock in Saudi Arabia, or ask them to move to Scandinavia. Move to Scandinavia. Forget the gene therapy. It's much cheaper. And it, so when you put things into this construct, a lot of things begin to make sense, and your way forward starts to fall together. Okay. I like this concept from Barton Childs and summary because it summarizes a number of things and pulls us back a little bit to think about things in terms of first principles. We've gotten very caught up in Oslerian medicine and its anatomical approach to disease. Consequently, we often view humans who are ill as broken machines and just needing another gear. My gear A is stripped, put another one in and everything will be fixed. What that avoids is forcing us to really look at the environment defined as the cultural, social, and true environmental situation that the person's living in. Whereas this evolutionary approach, championed by Barton, really forces us to be more inclusive and understanding of the entire family and the environment in addition to the genetics, and to look at genetics in the context of evolution. Another principle that comes out of this is that there are 3 lines of homeostasis that need to be considered when looking at undiagnosed disorders. One is the homeostasis of genetics, which arises out of evolutionary history. The second is the homeostasis of development, that is the developmental trajectory of the person. What were they exposed to? One of the disorders that I've personally studied, not at NIH, but previously at Baylor and at UBC, is Schmidt-Key immunoosseous dysplasia. Rare disorder, but we had considered it 100% lethal by early adulthood, until we began to sequence siblings of these children. And the reason we did this was because no model organism I ever made got ill. I made all these knockout mice and flies, and they were just fine. So we said, well, I wonder if, any children. I have siblings who have this. Yeah, sure enough, they had biallelic mutations, they were perfectly fine, and their sib was dead or very ill. So what does this suggest to us? That it is in the interaction with the environment, or their developmental history, and possibly their genetic background, that are contributing to penetrance of an autosomal recessive lethal disorder. The same would apply to PKU, which we were debating this morning. If you don't eat phenylalanine, you're never going to get PKU. Okay, so this then raises the challenge of evolutionary or genetic individuality and how we deal with this in terms of undiagnosed diseases. So to keep it interesting for all of you clinicians, I'm going to present some cases to illustrate this. So we were referred a patient, siblings actually, 2 patients, from a very excellent geneticist, wanting to know if these kids had a novel disorder of gluconeogenesis because they had multiple hypoglycemic episodes. The structure of the family, the two girls, unaffected parents. The older had normal development through 18 months, then delays, hypoglycemia beginning at 20 months, progressive development of dysmorphic features and obesity, and her younger sibling, delayed development from birth, hypoglycemic episodes, no dysmorphism or obesity. The older on the left and the younger on the right. Any diagnoses?
From the audience, I can't say.
I would consider Beckwith Wiedemann.
Beckwith-Wiedemann for the left. I'm waiting for Christine to say what the diagnosis is for the person on the left. So the girl on the left was diagnosed clinically with Smith-Magenis. And she ended up having a premature stop codon in RAI1. So, but her sister had no such mutation. And of course her parents were unaffected, so they didn't have it, so this had to arise de novo in her. But since when did Smith-Magenis syndrome cause hypoglycemia? Common feature of Smith-Magenis, right? No. So we performed exome sequencing, and they both had mutations in PCK1, which is the cytoplasmic PepCK. The mutation was in a beta-pleated sheet, destabilized the folding of the protein, so they had very low amounts of the protein. Well, that still begs the question then, since that doesn't account for developmental delay, of what's wrong with the younger child, unless you say she got a tremendous hypoglycemic hit, but there was no history of that.
Did the older one have any other symptoms of Smith-Magenis, clinical symptoms other than, to make you think about that? The things we discussed this morning?
She was relatively mild compared to the standard, the common deletion, right? So yeah, she had some of the other features, which was why the clinicians thought about it. The younger child had a de novo mutation in GRIN2B, which is a known cause of developmental delay. It's one of the subunits of the NMDA receptor. So what do you have here? Undiagnosed disease. Why? Because there's 3 diseases in the family. 2 de novo and 1 shared as a recessive disorder. So how are you going to approach this is really the question. These are your undiagnosed children in your clinic or adults. And so you have to be very, very systematic in your approach. Typological thinking no longer works. It is broken. Oslerian medicine fails at this part of individuality. So you might as well jettison all your medical thinking and all your medical training because it's useless.
Can I ask you,
Yeah.
new mutation or a known mutation?
It was a new mutation, and what I'm showing, I didn't go through the slide, but we did all the channel work in collaboration with Stephen Trinellis here at Emory. So Steve did what you see here is that you're looking at the current through the NMDA receptor when it's expressed in Xenopus oocytes, and what you observe is that there's much more current or reduced current in the mutant compared to the wild type when you add the glutamate.
So basically you did functional studies too.
Right.
Thank you.
So it was a rare de novo mutation, so we picked it up based on rarity and being de novo in a gene that we knew would cause developmental delay, and then asked Steve to do the functional studies on it.
Is it a highly consanguineous family or not?
No, this is an outbred family, no consanguinity. Okay. This is one example of something that led us to address an issue of how do you approach exome analysis. So human geneticists in genomics have fascinated me for quite some time. You realize they're the only geneticists that consider a diploid organism haploid. They have one reference sequence. We're not fungus, I mean, that's why the early geneticists used yeast and, all the different fungi, because they were great for genetic studies. Well, we put up one reference sequence. Go to NCBI. Where do you ever find the second strand to the reference sequence? It's just one. Well, you have an intrinsic problem. You have a diploid human that you're trying to align and annotate and genotype relative to a haploid average human. Doesn't work so well. So what you really need to do is align the sequence to the parents who are unaffected. What are you doing trying to align to this average human? Is that average human Chinese or Japanese or Korean or some random North American? Who is that average human? And how related to that person is the person you're looking at? This really arises out of fundamental biology, that this is a flawed concept. We develop new variation every meiosis. How many meioses away from that average human is the person you're looking at? And now you're going to try to align 100 base pair of reads to that average human and hope you don't have too many mismatches so that you can actually get those reads aligned? Well, I'm surprised it works as well as it does. And we actually make some progress with it. But, it's fundamentally flawed in its logic. So, what one would ideally want to do is sequence 23 linear chromosomes, globally aligned, chromosomes to reference, identify differences, and determine the causal variant. And this is where we've struggled for a few years now in terms of next generation sequencing. The real world is that you get millions of these short reads from 2 different copies of each chromosome, assemble and align reads, mixing up which is which, identify differences, most of which are false positives, determine causal difference, or mostly fail. Well, we fell into the category of mostly fail. We fell in so deep, I wasn't sure this program was going to keep going much farther. We needed a solution. And just aligning to the reference sequence wasn't cutting it. So what we came up with was the following cost-effective approach. That is, we ran SNP chips to detect CNVs and regions of homozygosity and chromosome mosaicism anyway. So, because the evolution of humans, the expansion of the human population has been so recent, what do you have left? Lots and lots of haploblocks. And you can impute those haploblocks from the SNP calls on a good SNP chip. So you can impute the parental references from their SNP chips. Impute those also sequence those individuals so you can refine their sequence with your exome sequence, then align the proband to those parental reference sequences. Now, why has no one ever done that before? Well, there's one fundamental problem. It's this thing right here called the liftover. You realize that you now, once you've built parental references, you have two unique genomes that are not annotated. So you can never tell whether a variant is deleterious or not deleterious or what genome or what gene it occurred in, because only the reference sequence is annotated. So you have to lift these two back over on top, the two parentals back over on top of each other, and then align them again to the reference sequence, which is annotated. So that was the work of a brilliant medical student who came for 4 months to work at the UDP. He's the most amazing fellow I've ever met in my life as a medical student. He walked in, he looked at it, and he said, "This is solvable." Sat down and wrote the code. Took us another year to get all the bumps smoothed out of it, but it's working now. So that was the sort of an illustration of the individuality component, and what does this mean then for what you can do that you weren't able to do before. What you're looking at here is the HLA region for a family. And any of you who have been involved in next generation sequencing, you know that all the molecular labs avoid the HLA region like the plague because they can't align it. They cannot call variants in it. And what you're seeing in the top two panels is the alignments that we got back from, or top three panels, from the intramural sequencing core at NIH. You see lots of variation in there. Now, you drop down to these middle panels here, and you see the alignment from the diploid aligner, as we call it. That variation has disappeared. The reads went to where they were supposed to. So now you could actually call mutations in the HLA region. So autoimmune diseases are now tractable. Okay. What other problem do you run, conceptual problem does one run into when you're doing these type of studies? We have one patient in front of us, not a cohort. How are we going to determine if patients are having this much variation just as a matter of being alive and the product of sex, which mutation did it? Or did any mutation do it? So what have we used in medical genetics traditionally? Well, let's back up and say, what do geneticists use traditionally to prove causation? Now, I'll tell you, this has never been published. It's an oral tradition that comes down from Euler and the early geneticists, but they never published a fundamental paper on the determination of causation for a genetic mutation. But the oral tradition is 1, independent alleles giving the same phenotype, 2, segregation with disease, and 3, rescue. Failure to meet those 3 criteria, you have not proven causation to a classical geneticist. We could never do that in human genetics or medical genetics because A, we can't breed our families. 2, we collect, our independent alleles were the collection of cohorts. And 3, we couldn't rescue. Gene therapy hasn't worked to this point, so how do you rescue? So what we really used was association power. But when you have an N of 1, you have no association power. So what are you going to prove? Prove that the mutation affected the protein? Yeah, okay. You proved protein pathology. What relevance is that to the person's disease? You know, maybe you can live without that protein. How do you know? How do you know whether it did or didn't cause the person's disease? Well, maybe that mutant protein also affects the cultured cell that you have from the patient. But again, how do you know whether it's relevant to the patient's disease or not? Then you can go on to knock it out in a model organism or do whatever you want to do, and hope that it recapitulates the disease. So have faith. Anyway, this really called into question a lot of what we were doing in the undiagnosed diseases program and the naivete with which we began. So this has required an extraordinary amount of thought and patience on how to address this issue. So you could say we could go back to association by building databases and seeing if we take all the unusual people in the world and pool them together into large databases, can we end up with multiple individuals with the same phenotype and mutations in the same gene? We are working on that. We're working on that in collaboration with the Canadian Forge Consortium and the European Union, and we've also been requested to deposit our data as we get this sorted out into dbGaP, which is where I presume also all of the extramural sites will be asked to deposit their data. But what would be another way forward? There's no philosophical breakthrough in dumping stuff in a database. Is there a way forward? So I'm going to give an example from one of the postdocs and one of the physicians in the undiagnosed disease program that suggests a way forward for some disorders. This is the family. The oldest individuals are actually deceased and not really shown that way on this pedigree. It's an autosomal dominant distal myopathy, and what they have is a very unusual distal myopathy in which the gastrocs become full of fat as the muscle tissue deteriorates, and you see the increased signal on the MRI where the fat has replaced the muscle tissue. So on biopsy, one sees these inclusions in the muscle tissue. So it's an inclusion distal myopathy. So who done it? These individuals did not have any mutations in the known causes of inclusion myopathy. And in the process of sequencing and aligning, what I noticed when reading the next-gen sequence was right in this exon here, there were 2 missense mutations. And when we did the Sanger sequencing to confirm that, what we found was a deletion in the intron. Which, as a biologist, it takes you a second to look at, and you say, that's the lariat acceptor site, I bet this splicing screwed up. And indeed, that's what we showed. But the gene was HINT3, HINT3. And I bet you all heard of it too. Nobody's ever studied it. So, did this gene do it? Could it have done it? I mean, it's a gene never studied. It's a distal myopathy never described. Well, it altered splicing. So we made an antibody to HINT3, and indeed it's in the muscle inclusions. But did it do it? it's in the inclusion, but was it just captured there, sequestered there by chance? Or is it actually the seed crystal for the formation of the inclusion? What would happen if we express that in cultured cells? Oh, indeed, we get aggregates. So maybe it done did it. And when we get those aggregates, we don't, with the mutant form, this deleted form of the hint tree, the cells die. Whereas if we express the wild type protein, they don't. So how are they dying? Endoplasmic stress, endoplasmic reticulum stress, unfolded protein response leading to apoptosis. Well, we said we have a new cause of distal myopathy. So we sequenced 500 other random patients with distal myopathy and never found another mutation in this gene. Does that matter? No. Why?
You're comparing apples to oranges.
Right, but you still haven't proven that it's causing the disease, even though you have segregation of the mutation with it and a possible mechanism. What we went back and did is look at all these other inclusion distal myopathies and say, what mechanism are they leading to cell death by? ER stress and apoptosis. So what was conserved is the mechanism of cell death. The causative protein is irrelevant. Why would one expect another HINT3 patient to exist in the world? You don't. If the protein can form inclusions and cause ER stress, it is quite capable of giving the disease. So it doesn't matter what the protein is. So the association now is at the level of a systems biology approach and not at the level of the gene. So there is a way forward in causation if you can relate to a shared mechanism of disease. So in this case, what constituted the evidence for disease or of causation? It's again association, but from a different perspective. We went on to make zebrafish that express the protein and they have muscle troubles. Okay, what was the other problem with the undiagnosed diseases program? Is that we were awash in patients and in great dearth of solutions. How has traditionally translational medicine worked? Smart Dr. A, who either has a very close colleague who's a PhD or also has a research endeavor, generates hypothesis A when he sees the patient and walks over to the lab and says to graduate student or postdoc, could you test this hypothesis? The person tests it and it works, it validates or doesn't validate, but what it inevitably does is refine the hypothesis. So there's an iterative process of calling the patient back and then testing that new hypothesis from the lab. On the patient, hypothesis is refined, and you go back to the lab. You ever tried doing that for 200 a year yourself? 200 different diseases a year, not just one. There's something seriously troubling here. Welcome if you're applying for an undiagnosed diseases program. Think about how you're going to deal with this. Because otherwise you have a big basket full of things, like I did. So this is what I described, sort of this iterative circle that people go through in translational medicine. And hopefully, it goes all the way around to actually give some therapy. What did we end up having to come up with? So I would like to go back to the point that I raised early on. This iterative hypothesis generation and testing is constructed on our known knowledge and our known insights. It's an Oslerian approach, not one that Archibald Garrett would have been terribly thrilled with. So I had to think up, with the permission of Bill and the tolerance of the others, an alternate approach. And it's really this. How can we clinically phenotype standardized? The letters from each clinician have to be identical in their description of the disorder. They cannot vary. If it's midface hypoplasia, it's midface hypoplasia, and both mean the same thing. So, second, it has to be discrete and quantitative so I can use it in a computer. It has to feed into bioinformatics. Why do I want to do this? We are all subject to anecdotal bias. The stone I found my most recent reward under is the one I am always going to guess first. That was an absolute death knell to the program at NIH. And I cannot tell you how hard it was to get the physicians to leave their ego in the closet. You'll struggle with the same thing. People are people everywhere. So this clinical phenotyping needs to be divided into two components. One subjective, my subjective interpretation of what's going on. The other discrete and objective. And that which is discrete and objective must be performed in an ontology. We chose to use human phenotype ontology. Why must it be in an ontology? The ontologies have been mapped across species. That means, and they've been mapped to every human disease. So the program the physicians use now, as they map the phenotype, it tells them all known human diseases with those characteristics, plus it weights other physical features that are commonly found and says, did you look for this? So it leads to a very thorough phenotyping, and as they go, we on the fly search every known mutant organism of 6 to say these are the candidate genes that could give rise to this combination of phenotypic features. Biochemical phenotyping. Why do we need to do that? And we need to do it untargeted, agnostic. We have no clue where the disturbance of homeostasis is. If we can identify it by biochemical phenotyping, we can then integrate that with the clinical phenotyping and the genetic phenotyping, which is the SNP chips and whole genome or whole exome sequencing. This then all feeds together in a bioinformatic analysis that's computational. No human involved. This data is used to weight the variance. Not human guessing, not human bias. Here is where the human bias returns. Now all this data feeds back to the clinician, and they can use their subjective interpretation of what they observe to say, of these weighted variants, these are the ones we consider most likely to be causative of the disease. Now, it was completely unaffordable, despite the large amount of money given to the UDP, to get much farther than this in any constructive way. And we haven't really given much back to the patient yet. You notice that? So you have these candidate genes that will come out of this. So one of the first things that Bill and I requested was the ability to get these candidate genes out to the extramural environment so that experts could look at these, study these, and say, could this gene cause or not cause the disease? So that's this gene function list, and you guys have probably seen these RFAs for supplements to R01s, and the last RFA was for an R21, and then there should be an RFA for R01s coming out in the fall. The alternative, and then also deposit this data in the database, as I described. And then the alternative is, well, this is such a good candidate, and we could really leverage a lot of collaborative help if we made a model organism. Now, I've avoided mice altogether, but flies, yeast, and zebrafish, yeah, we'll make those. Okay. So that's the model that we're working with currently in terms of trying to parallelize translational medicine. I went over this. The biochemical phenotyping that we're doing right now is metabolomics, lipidomics, glycomics, and energetics. Okay, a couple minutes left. Now I want to tell you something, just for fun. Because it's kind of cool. It's one of Dr. B's cases that she helped solve. So this is the family, and you all here identified mutations in this gene. Now, these two children are late childhood, 10, 12 years old. The only other case ever reported before died as a neonate. Why did these guys survive? Well, milder mutations, right? Well, it has premature stop for one, and has two conserved amino acids mutated in the other. And there wasn't much residual activity in the cells. So maybe it wasn't milder. And when they ran a Western on the cells from the patient, there was no detectable protein. Maybe it wasn't milder. So why did they survive? Well, I'll make a long story short, and I won't tell you how I got there. Check out the blood genotype. Oh, and here's Meow's lab staining with Con-A, showing that the patients, all their cells have this abnormal glycosylation on their surface. So now, check out the blood genotypes. How many do you see in the two siblings? 8. They have 8 genotypes in their peripheral blood. What came out of the DNA diagnostic lab and when we sequenced their blood by Sanger? Heterozygosity and segregation of mutations. The reason they're alive is intragenic, mitotic recombination. You see? Both of them have wild-type alleles. So let me explain. These two mutations come from dad. This one comes from mom. This is the premature stop. So if you look at that and you say, okay, this is what we would expect for the allele from dad, because we have wild type for mom and mutant for dad, right? Got the mutant paternal allele. And then down here, this is what we would expect from mom. Mutant for mom and wild type where dad is mutated. Well, how do you get these? Right? Mutant for dad, mutant for mom, and a wild type for dad. Mutant for dad, wild type for dad, wild type for mom. Wild type for mom, Mutant for dad, wild type for dad. Mutant for dad, mutant for dad, mutant for mom. Wild type, wild type, wild type. Again, no wonder the kids are undiagnosed. You would have expected an embryonic lethal or a neonatal lethal with very severe features. They have the biochemical features of the disease on Meow's studies, but they don't fit the Oslerian typological classification of the disorder. Evolution is at work at the cellular level here. There is a strong selective pressure for a wild type allele. That's it. I'd like to acknowledge all the people at the NIH who've contributed to this and made it possible for me to pursue these studies, and I'll leave you with a takeaway quote.
So you're showing a significant amount of functional studies after, like to establish causation. When exome analysis is so widely being available now, where do you think the rest of the physicians who are ordering exome should go? Like when you get all these variants of unknown significance, what do you think?
Well, they're of unknown significance because we haven't looked at them, right?
Right.
You can't get by the functional studies. End of the discussion. There is no, to my mind, there's no functional there is no philosophical breakthrough. And it's going to become increasingly more difficult as you really think about this. We've only scratched the surface. Because, we're still thinking in a one gene done it mode. Even when I present, 3 disorders in this family, right? I broke it up into 3 disorders and 1 gene done it. We really haven't systematically analyzed next generation sequence in terms of the combinations, let alone the ecological niche that's necessary for expression of the phenotype. So, and we're not going to get there very rapidly because it is a very difficult problem. But we will never get there if we're not extremely systematic in our approach. And that was the hardest mountain to climb, was to convince people to be systematic. The advantage that you have setting up such a clinic or a program here, if you did, is to start out that way, knowing at least a minimum of this, the systematic approach that you need to take. So metabolomics, how long, what's most people's experience with that? It reflects better the person who froze down the CSF or something than it does the disease at hand. So it's being very precise and systematic about what you did. That's the way forward. Then it just becomes a lot of fun math and computation. And, and that's the fun of having an undergraduate university. You have math majors, you have CS majors, and you feed them your ideas and let them run and put them on the supercomputing cluster. And it's great.
Among the 3 phenotypes you showed, the clinical phenotype,
Yeah.
the metabolic, and the genotype, you probably will have congruity in the genotype as well as the biochemical. But when it comes to clinical phenotype, harmonizing and normalizing the data is extremely difficult because each one will have their completely different interpretations. HPO, the human phenotype ontology, has few terms. And so how are you going to approach, how are you going to get a really accurate representation of, well, it's hard to say what is normal and what is abnormal. It's difficult to define what is normal. So how are you going to harmonize the clinical phenotype data for further bioinformatics analysis? Or what's your approach?
So our approach was we're working with the programmers who began developing this for the Canadian consortium in Toronto. They developed this freeware called Phenotypes, which I would recommend that you take a look at. I sort of found out about it backwards, which was a little bit of an embarrassment to me because, I'm from UBC and, I think I knew what the Canadians were doing. And but my friend from Berlin called me and he said, have you seen this program? We just put it in the medical genetics clinic. It is the best thing ever. And that was Peter Robinson, who actually founded HPO and is still curating it. And this is an interface which is very straightforward for clinicians to use. And because it shows you the hierarchy, right? So you could pick ataxia, and another person might pick some subset of ataxia that's under it. So when you go to do your bioinformatic analysis, because you know that it's in the hierarchy, just because one person said simply ataxia and one said a more specific form, they're still linked, right? So that's getting at one of the issues of whether one physician was as refined a neurologist as another or refined a dysmorphologist as another. It's still in the same hierarchy. The second is NIH has funded this consortium from Berkeley and Portland and Pittsburgh and who knows all where, called the Monarch Consortium to map the HPO terms across model organisms and to human diseases. So my issue was not really which ontology to use, but what interface to use that would make it possible for the clinical staff to easily, rapidly map into HPO. We had started out using MedGen, which was developed by the National Library of Medicine as an ontology and to map much more broadly. And the problem was no interface. And the clinicians absolutely rebelled and nothing was going to get done. Whereas with the phenotypes, there's several advantages built in, nice things built into it. One, you can draw your pedigrees in. Two, it tells you other physical features that you could have looked for. And it also, on the fly, maps to all the Ullmann diseases. And then once this candidate gene part is fully operating, there will be additional positive feedback for physicians because then you can roll right over to the exome data and say, are there mutations in any of those genes? So, is everything perfect and seamless yet? No. This is really a work in progress in which, when, Congress and Obama finally said, okay, they would spend some money, that's when we issued the contracts to try to smooth out some of these bumps in this analysis and bring these things up to the level that we thought would be useful for the extramural centers as well as ourselves.
So in your informatics pipeline, how many patients fall out where it's obvious what the diagnosis is, what the mutation is early on without having to move on? I just want to get a sense of, how much is we just deal with rare things, so they may not get a diagnosis because they hadn't seen the right physician versus this really is an unknown that needs more functional analysis.
For us, it's probably around 5%. It's very low. Now, that will largely depend on for the extramural sites, that will probably be slightly different. But, our patients have been to you guys. Then they've been to Mayo. And then they've been to the Cleveland Clinic. And then they end up, that was my argument with the clinicians. I was like, what makes you think you're brighter than the guys they went to see? You're all looking in the same textbook. So it's like, time out. We have to have another approach. And so, you know, it would largely be how you constructed the undiagnosed disease clinic here. If you really put it at the end of them having seen the bright people that should have made the diagnosis, you're going to have a very low pickup rate.
Thank you for wonderful talk. So look at the last case about CDG2B, right? So we are looking at almost like somatic gene adaptation. So my question is, if that is more frequent than we thought, do you think you can see those by whole exome sequencing and go through your data in other diagnosed patients and see whether similar observation exists?
Well, it's probably more frequent than we realize, but we got lucky here in being able to pick this up because because we could PCR across all 3 mutations and get it on one strand of DNA. So when we did single-strand DNA sequencing, we could pick that up, right? You're not going to have that advantage very often in next generation. Certainly next generation sequencing wouldn't have picked this up because they're too far apart. Because you have 100 base pair reads, and you're trying to put those together. So unless you have other anchoring SNPs adjacent, you won't be able to arrange your single base pair reads to say they all came from the same allele. Whereas we could do that with PCR and then cloning afterward. So I don't think we're going to be easily pick this up by next generation. It's just going to be one of those things where you decide to pursue something because it doesn't make sense. And so to make a long story short, how we ended up here was, why was there no protein in the patients, right? So you have to have some, we only had one stop codon for nonsense mediated RNA decay. What ended up happening is that the splicing from the first exon to the splicing from this first exon to the second exon is weak in wild type gene. This mutation, R110H, is very close to the splice donor and appears to make it splice even less well, so that you get read-through into the intron, and that results in a premature stop. So that you have nonsense-mediated RNA decay for both alleles, both the maternal, which had the premature stop, and the paternal, which presumably only had missense. That in the process of doing that, we found these cDNAs that didn't have this mutation, R110H. So I said to the postdoc, not buying your story, what's going on? Okay, that is the long story for how we got there.
I was thinking about whether that caused any significant change of percentage of wild type allele.
Well, I mean, we could call the patients back now and repeat the analysis on their blood DNA now and say, over the course of X number of years since you first came to the UDP, what is the predominance of wild type allele?
That's one of the things that relatively common in CDG is the changes, glycosylation changes in the blood often improve with time. So maybe this is one of the reasons.
Right. Well, and what I guess to expand on your point is the fibroblast cell line that was established in the UDP prior to my arrival is not really mosaic. So this is a blood phenomenon.
I just have a question. How many cases do you get from outside America? Because I believe that, American population is not as consanguineous as Middle East population, for example. And I believe that the greatest challenge you'll be faced is by cases from Middle East, like Saudi Arabia, for example. So are you really presenting yourself outside USA? Do you think you are presenting yourself as NIH and diagnosis?
The answer is not that many people are from outside the United States, but some are. So, the first obligation is to U.S. taxpayers. And the NIH doesn't accept fee-for-service. So it's not so we have to avoid a congressional inquiry, which is usually the first thing you avoid when you're at NIH. And so that means making certain that you're taking at least a reasonable number of U.S. citizens. But are there others from other countries? The answer is yes. The consanguinity issue, no one has addressed in any way, period. Almost certainly, the researchers who are doing the work in consanguinity are stopping at the first mutation and calling it quits in a day in a paper. And the few cases that we've taken with consanguinity, we did the same, and we right well know that there are many, many additional mutations that are modifying this. But remember, papers are not truth, they're stories. And you need a certain number of stories a year to justify your existence. And they're not false, but they're the story that explains, in part, the information. The problem we lack is combinatorial genetics in model organisms. It's really difficult to breed in or introduce in a short time frame multiple genetics and look at the interactions.
Thank you.